Well, as we let other people come in, let's start so we keep on schedule. Delighted to welcome everyone to a repeat performance. Both Roger and Alan were here about this time last year when things weren't, shall we say, looking quite as promising as they do now. And hats off, really a big thank you to Alan and Roger and the teams that they've been involved in. Obviously, it's not just the two of them, but the teams that they've been involved with and the other groups out there. A massive problem, as we all know, confronted us 15 months ago. And I think none of us could have predicted how well science responded and the companies and the academic labs responded to the challenge. And by working essentially nonstop, I think I'm correct in this. I don't think Alan and Roger have slept much the last 15 months. They'll probably sleep maybe a year from now, but we do owe them a debt of gratitude. And again, I thank you both for that. So they've graciously carved some time out of their schedules, which are still pretty hectic, it sounds like, to kind of give us an update on what we've learned in the last year and where we might be headed. And again, they don't have crystal balls, but they definitely have their finger to the pulse of what's happening with COVID vaccines. So Alan Barrett, director of the Seeley Center Institute for Vaccine Sciences at UTMB, and number of appointments with the World Health Organization. I won't go through his achievements. It's on his bio. Will lead us off, and then Roger Nosal, a good friend, who's head of or VP, head of global chemistry, manufacturing and controls at Pfizer, will follow up. Questions we'll take to both of them or general at the end. So they'll both, as we do, will speak for a half hour, and questions we'll pick out of the chat, or most likely we'll just have you unmute and ask your questions. Okay, so with no further ado, I am going to mute myself and go off video and turn it over to Alan, and then Roger will come on in about a half hour. So Alan, again, many thanks for your hard work the last year and the anticipated work that you'll be doing. And again, thanks for joining us.
Thank you very much, Dan. It's great to be here again, talking about COVID. A lot of people have done a lot of work in the last year, and what I'm going to try and do in my little session for the next 30, it's probably near 35 minutes, is try and give an overview of where we are now. To put it in perspective, there's over 200 papers published each day on COVID, and it's really very hard to keep up with what's going on. But what I'm going to try and do is give you an overview of the current situation of where we are now. Suzanne, do I have to launch my screen, or are you going to launch it for me?
You can launch it like we did in the practice. Just share your slides, share screen.
Okay, great. Okay, thank you.
And then go to full screen.
Yeah. So I'm going to give my usual disclaimer. I'm a director of a WHO Collaborating Center, and I'm also a member of the Scientific Advisory Committee for the Coalition for Epidemic Preparedness Innovation, CEPI. And CEPI is supporting 8 COVID vaccine candidates. And so I can have no opinion on any particular vaccine candidate for or against it, just so I can say that at the beginning. So what I'm going to try and do is give a paintbrush overview of where we are with different things. So where are we now in terms of vaccine candidates? We, as of May 25th, have 285 candidates that are there. In addition, there's another 50 to 70 in academic labs and small biotechs around the world. 101 have proceeded to clinical trials. And of that 101, 15 have become authorized or licensed around the world in different countries, including 3 in this country. And we'll talk more about those as we go through the talk. In terms of vaccine immunogens, various technologies have been used. You name it, it's been tried. So we have the traditional inactivated virus with and without one or more adjuvants. We have the entire spike protein, which can be in the pre or post-fusion form. And the difference here is that the post-fusion is as it's found in the cells, as after fusion. The pre-fusion is what's found in the virions. And of course, as you might imagine, the pre-fusion appears to be a better imagen than the post-fusion version. Of the spike protein, it's got different subunits, S1 and S2 being the big ones. S1 is thought to be the major imagen and includes a receptor binding domain, and there's various candidates based on these different imagens. While the spike protein is a major imagen that's used by the vast majority of the candidates, some are using different combinations with other structural proteins and envelope membrane nucleic capsid. And there are a few candidates using non-structural proteins or peptide imagens. So the big question, first of all, is do vaccines prevent COVID? And of course, I think we all know the answer is yes. All these studies to make this conclusion are based on vaccine efficacy studies from clinical trials. We have placebo controls, and you show that there are less cases of COVID in the vaccinated group as compared to the placebo. And we'll talk more about that as we go along. The mechanism of protection of the vaccines is not known and likely differs because the vaccine platforms are so varied. And so we can think of the vaccines being qualitatively different. Now, when you're in vaccinology, you want a correlate of protection. So you can use this as a marker to know whether your vaccine's working, and you can use it in the production, manufacture, and quality control of vaccines. But we have no correlate of protection established yet for any of the COVID vaccines. Many people believe that the correlate of protection is going to be neutralizing antibodies, but the evidence for that is not conclusive. There is no definitive neutralizing antibody titers associated with protection from disease. And we'll talk more about this as we go along. So we currently think of neutralizing antibodies as being the biomarker of protection, but it's not a correlate. And because of this, there are a number of clinical trials going on now with different vaccine candidates involved in what we call immunobridging, where they're trying to relate vaccine efficacy in terms of clinical evaluation compared to a correlate protection. And I would imagine we're going to get a lot of data in the next 6 months towards answering these questions. So if we turn to the clinical trials, I said at the beginning, there were 101 vaccines in clinical evaluation. Of those, 21 have been in clinical evaluation in the United States. And it's a variety of different platforms, as you might anticipate. There are 2 DNA vaccines, there are 4 RNA vaccines, there are no inactivated vaccines, there are 4 recombinant protein-derived vaccine candidates, there are 3 replicating viral vectors. I'm just going to make a point here because these platforms are very different. For the replicated viral vectors, Merck have used live vesicular stomatitis virus and live measles virus as vectors. And I'm going to not say this with MISA, I'm not sure I'm saying this properly. They're using a live respiratory syncytial virus vector vaccines. There are a number of non-replicating viral vectors, most of these based on adenoviruses or pox viruses. There are 7 of these currently in clinical evaluation. We have no live attenuated vaccines in the United States in evaluation. We have 1 based on virus-like particles, which is based on expression of VLPs in plants. And we have no peptide vaccine. So quite a variety of different platforms. So of those 21 that have been in clinical trials, they've had various success stories. And I'm going to try and focus on the ones that have either dropped out or the ones that are going forward. Inovio's DNA vaccine has not progressed from phase 2, and it's unclear whether they'll proceed to phase 3. As we all know, the Pfizer vaccine went for emergency use authorization, as did the Moderna vaccine, but there are a couple of messenger RNA vaccines currently being evaluated. In terms of recombinant protein, Novavax is thought to be getting near its application to the FDA. Sanofi collaborated with GSK. Sanofi had a recombinant protein together with GSK's ASO3 adjuvant. When this was first made, it didn't do very well in terms of immunogenicity in the over 50s. And so they've reformulated it, and the phase 2 trial looks promising, and they're now going into phase 3. But it's really going to be probably towards the end of this calendar year before we see any data on the phase 3s. In terms of replicating viral vectors, the two candidates made by Merck, the measles virus vector and the VSV vector, both of those dropped out. And the issue there for Merck was that the levels of neutralizing antibodies induced by these two candidates were not as good as convalescent plasma, and so they stopped studies. In terms of non-replicating viral vectors, the AstraZeneca has clinical trials being done in the US, and it's unclear when that's going to go to the FDA for approval. The Janssen vaccine has gone for approval. Vaxart was an adenovirus 5 vectored vaccine given orally, and that didn't do too well in the phase 1 clinical trials. The other ones, there is not really much data in the public domain to say how those are getting on at the current time. So when we think of how the vaccines are going to move forward to get approved in this country, the FDA licensed vaccines in the United States, but it's the CDC Advisory Committee on Immunization Practices that decides on how a vaccine is particularly implemented. And in the case of COVID, the issue was we could not go through the entire normal licensing procedure to get vaccines out there because of the time taken to do that. And an alternative approach has been used called emergency use authorization. And that's a scenario where a vaccine, a drug, or a diagnostic has a clear beneficial effect that makes its use warranted, even though the product is not licensed. It's clear benefit. This was first started in the Bush administration post the 2003 anthrax attack. They were decided there was a need to have some sort of emergency use when we would need diagnostics, vaccines, and drugs for particular scenarios. The EUA is something that's put out by the FDA. But it needs to be remembered, even though such authorizations can happen in emergency scenarios, once the emergency situation is over, then the procedure for licensure of the vaccine has to go back to the traditional routes before it can be licensed. And a good example of this was the Merck Ebola vaccine, which got an emergency use, but it took 3 years later before it actually got approved through the whole licensure process by the FDA. So as you all know, I'm sure there are 3 vaccines licensed in, authorized in this country. 2 of them are messenger RNA vaccines from Pfizer and Moderna. And the messenger RNA vaccines work on the principle that you have a messenger RNA encoding the particular gene, in this case a spike protein. You have it in a lipid nanoparticle. The lipid nanoparticle is then taken up into the cell, and then the RNA is released into the cytoplasm and gets translated to make the protein. And then you make an immune response against the protein. And that, as we know, has proved very efficacious. The RNA sequences are proprietary, and maybe Roger will talk about that, but I somehow doubt it. And in addition, the lipid nanoparticles are also proprietary. This is a slide I borrowed from a review showing that the actual components of the lipid nanoparticles can vary greatly. And so again, these are proprietary, how the messenger RNA is actually encapsulated within this lipid nanoparticle. And so we don't have much information on this for both Pfizer and the Moderna vaccines. The Pfizer vaccine was the first one to go to emergency use authorization. This was a pre-fusion stabilized membrane-anchored spike protein given as a 2-dose regimen 21 days apart. And when the results came out, it was really very impressive at 95% effective preventing COVID in terms of vaccine efficacy. And this was totally unexpected. Indeed, the FDA, when it gave its guidance documents out for what vaccine producers needed to do to get a vaccine through to an EUA, they were looking at something like 50% efficacy. And so when this result came out at 95, it was an amazing result to see. I think everybody could say that. And not only was this vaccine efficacious in adults, but it was also in senior citizens, 94%. And I remember at our site, we had our oldest vaccinee was 85. And so it's really a very impressive vaccine in terms of its efficacy. It got emergency use authorization in December 2020. It was approved for those aged 16 years and older, except for pregnant women. And then the follow-up in March of this year showed that 6 months later, the vaccine is still 91% efficacious in terms of vaccine efficacy. And against severe disease, it's 100%. So it looks as if the vaccine induces immune response that lasts at least for protection for 6 months. Pfizer followed this up with a number of studies done in children. The first one they put out in March was adolescents aged 12 to 15, and really surprisingly, the efficacy was 100%. I don't think people expected that again, but it looked really good. And the antibody responses were really very strong. And one of the things that has been seen with the vaccines, overall, I'm paintbrushing here, is that the immune response for the vaccines gets weaker as you get older, which is what you see with vaccines. But interestingly, in terms of natural infections, you normally see higher neutralizing antibody responses in older people as compared to younger people. And maybe we can talk about that later on. The FDA approved the modification to the EUA in May the 7th, and this was then endorsed by the CDC on May the 12th. There's a follow-up taking place now. Pfizer and as the other companies have got studies going down to younger ages. So now we've got approval from 12 and older. There are 3 different groups that are consecutively being analyzed for immunity: 5 to 11, 2 to 5, and 6 months to 2 years. And I think this is quite exciting in terms of the potential of school-age children being able to go back to school fully vaccinated in the fall of this year. Maybe I'm being a bit optimistic. We were asked to do some crystal ball gazing by Stan, and I'm hopeful that we can actually have all our school children going back to school in the fall. The Moderna vaccine was also equally very impressive. It gave 94.5% efficacy. And there was a remarkable, they went from 65 days from getting the nucleotide sequence to the phase 1 clinical trial starting, which is absolutely amazing. Their vaccine was given an emergency use authorization the week after the Pfizer vaccine, and has been approved for those 18 years and older. Just as with the Pfizer vaccine, in April, Moderna reported their vaccine was also 90% effective at preventing COVID 6 months after immunization. So again, we have the 2 vaccines showing pretty impressive immune responses and protection rates. And recently, just as with Pfizer, Moderna followed up showing that their vaccine has 100% efficacy in ages 12 to 17. So I think we're in a very strong position with the vaccines for different ages. The other vaccine candidate approved in the United States is the Janssen vaccine, and this is a replication defective human adenovirus 26 vector vaccine. And the idea was here that the human adenovirus 26 is very rare, and it could be used as a candidate vaccine because there'll be a lack of immunity in the population. This is based on the so-called AdVac viral vector technology from Janssen. It's been used for their Ebola vaccine, which has been approved in a number of countries and has been used for a number of other different vaccines in clinical trials. So again, this was the entire spike protein put onto the Ad26 virus. And unlike the other 2 vaccines, this is a single dose regimen. And this study was one of the first to describe differences in geography in terms of the vaccine efficacy. They described the vaccine efficacy as 67% effective overall, with an 85% effectiveness against severe COVID. And interestingly, in the United States, the vaccine was 72% vaccine efficacy, 66% in Latin America, and 57% in South Africa. And as we now know, there are different genetic variants in different countries, and the vaccines vary in their efficacy depending which of these genetic variants we're looking at. And this vaccine had its emergency use authorization in February the 27th this year. Alongside this, the Oxford have developed a defective chimpanzee adenovirus vector platform. The idea being that there's no immunity in humans to chimpanzee adenoviruses. They have a 2-dose regimen. They've done a phase 3 in multiple countries. It gives pretty good immunogenicity. And the US trial reported in March 22nd is a 79% efficacy, including 80% in the over 65s. But this one, so far, there's no information about it proceeding to emergency use authorization. So I think when we think of vaccines, we always think about, we want a vaccine that is not only 100% efficacious, we want it to be 150% safe. And so there's been a lot of interest in potential safety issues associated with the vaccines. The first one that came up was anaphylaxis, and there'd been initially some studies in Europe suggesting anaphylaxis was associated with some of the vaccines. The CDC did a follow-up of the Pfizer vaccine that got reported early January when they looked at the first 2 million doses of the Pfizer vaccine, dose 1. They identified adverse events. This is all adverse events, not just severe everything. And they got 0.2% of vaccinees describing adverse events, like anything from a sore arm to anything else. There were 21 adverse events associated with anaphylaxis, which was 11.1 per million doses administered. Whereas with licensed vaccines, it's an average about 1 per million doses distributed. Of these 21 adverse events, 17 had a history of allergies or allergic reactions, and 7 had a history of anaphylaxis. And the average time to these symptoms was 13 minutes after receiving the vaccine. So clearly more studies are needed. And you may ask, well, why wasn't it seen in the clinical trials? And clinical trials tend to focus on healthy people and special populations as we go on. And so people with allergies were excluded from the phase 3 clinical trials. And so that's why it wasn't seen in the clinical trial. There's been lots of discussion about what causes the anaphylaxis, and people have been looking at the polyethylene glycol component of the lipid nanoparticles. However, at the moment, the vaccine is contraindicated for those who have allergies, not surprisingly. The Moderna vaccine showed somewhat similar data by the CDC. They had 4 million people who had received the vaccine, and they found that the adverse events reporting was 0.03%. There were 10 adverse events associated with anaphylaxis, 2.5 per million doses administered. And of these 10, 9 had a history of allergies or allergic reactions, and 5 had a history of anaphylaxis. And just as with the Pfizer vaccine, it was fairly rapid. This took place after vaccination at 7.5 minutes. For the Moderna one, there's a suggestion it may be gender linked, with it being many more women than men showing the anaphylaxis. But I think more data is needed because many of the people who first got the vaccine were healthcare workers, and many of these are women, in particular nursing. And so I think the push towards gender has to be proven yet. And again, the clinical trials didn't involve those with allergies, and so it wasn't seen in the clinical trials. And people again are still looking at the polyethylene glycol excipient. Now, just to go back to a slide I showed you earlier, the LNP formulation involves polyethylene glycol, and so there is some suspicion this may be involved, but nothing is proven at this present time, to my knowledge, about which component is causing the anaphylaxis. After this, more recently, in Europe, the European Medicines Agency has reported an unusual thrombosis called cerebral venous sinus thrombosis, CVST, and has been seen with the adenovirus vectored vaccines. They have described it seen with the Janssen vaccine. It's been somewhat unusual that the thrombosis is seen in veins in the brain and the abdomen and arteries, and it's been associated with low levels of platelets and sometimes bleeding. The EMA in Europe concluded that there should be a warning on the vaccine about unusual blood clots with low blood platelets on the information for the vaccine. But critically, I think it's important to note they thought this was a very rare side effect of the vaccine and stress the overall benefits of the COVID vaccine from Janssen greatly outweighs the risks of the side effects. And a later study put out by EMA put the rate for the AstraZeneca chimpanzee adenovirus vector vaccine at 5 per million for CVST versus COVID cases, so thrombosis, somewhere between 39 and 48 per million. United States CDC investigated it, and we call it something different over here, TTS. And when the evaluation was done of different vaccines, it was found in April of the 135 million doses of Pfizer vaccine administered, there were no cases reported. Of the 110 million doses of Moderna vaccine administered, only 3 cases. However, with the Janssen vaccine, they identified 28 cases in 8 million doses, suggesting the risk is associated with the adenovirus vectored vaccines. we look at the data for the Janssen vaccine, they had roughly 8 million doses. As of April, there were 15 confirmed cases of TTS, and the average time of onset of the clinical symptoms was at 8 days. And 12 of the 15 cases had CVST, and 10 of the 15 had low platelet levels. Interestingly, it was biased towards women. In the ages 18 to 49, there were no men, nor there were any men in the over 50s. But when you looked at women, it was predominantly in younger women, aged 18 to 49, with the average age 37 for the cases, whereas much lower in women over the age of 50. The vaccine was stopped for Janssen while the investigation was done before it was restarted, and CDC have now data as of May the 13th that there are now 28 cases being reported, and including 6 males. And only one of these cases has unfortunately died from having the vaccine and this clinical condition. Most recently, myocarditis has been associated with the messenger RNA vaccines, and this was discussed by ACIP's COVID Vaccine Safety Technical Group, which meets weekly. And at the moment, it's just looking at very rare reports of myocarditis. They appear to take place in young people, more often in males than females, more often following dose 2 than dose 1, and very early after vaccination. But a lot more needs to be found out if this is a problem or not. And so far, the data indicates these myocarditis cases are mild and occurring in a very low number of people. So in addition to the safety issue of side effects, the other question is, can you still get infected if you've had the vaccine? And a very elegant study by CDC, where they followed up individuals and healthcare workers, as well as people in the general population. And they found that in the first 95 million doses of the 3 authorized vaccines, there were only 0.01% of people who had evidence of getting a COVID infection. And most of these people didn't have clinical symptoms, and only one person has died out of this 95 million. And 20 of the, 1% were breakthrough infections. 20 people have died, but they don't appear to be related to COVID-19. There are other reasons why these people have passed away. Other studies have looked at the healthcare workers to ask the question whether people who are protected from it, do they still get infected, and can they infect others? And the data is really very impressive, showing that the vaccines appear to prevent infection and the ability of vaccinated people to spread the virus to others. So the vaccines look very good at the current time. Now, if we turn over to the world, we have actually 15 different vaccines in either approved or authorized, depending on the country. We have 2 messenger RNA vaccines we talked about, Pfizer and Moderna. We have 3 adenovirus vectored vaccines, Janssen and AstraZeneca we've talked about, but there's also one in Russia, which is a heterologous prime boost, where you go in with adenovirus 26 and boost with adenovirus type 5. There is one protein-based vaccine in China. There is also one live attenuated vaccine that's been made in Russia, and this has been based on codon deoptimization to make a live attenuated vaccine given by the intranasal route. But there is no published studies on this to actually look at vaccine efficacy. There are a number of inactivated vaccines I've shown in blue here, and there is one vaccine from Vector, which is a research lab in Novosibirsk, which is based on SARS-CoV-2 peptides, and it's all proprietary. They don't tell you what the peptide sequences are. So there are a number of vaccines that have been approved, but actually there's a paucity of data on these vaccines in the public domain to know how effective they are at preventing COVID disease. And if you look at what's out there in the public domain, it can really be put down to 3 of the platforms. The data I showed you earlier for the messenger RNA vaccines for Pfizer and Moderna is very impressive, 94 and 95%. The inactivated vaccines, there's been data produced for 2 of the Chinese and the Indian vaccine, and they appear to have slightly lower vaccine efficacy based on these trials of 78, 78, and maybe as low as 50% from one of the Chinese inactivated vaccines. The Janssen vaccine is around about 66% efficacy. The heterologous prime boost vaccine, Sputnik 5 from Russia, is 91%. And the chimpanzee adenovirus vector vaccine AstraZeneca is around about 62%. Now, I've highlighted it and put it in question mark because there's been a number of different clinical trials with this. Each trial varies with the spacing between the first and second dose of vaccine. And the data for AstraZeneca is indicative that if you increase the spacing between the first and second dose, they could be more efficacious. But that data is based on relatively small numbers of people. All the vaccines that have been reported data so far are 2-dose regimens, except for Janssen. And on the immunity measured for these percentage vaccine efficacy I'm showing here are usually 1 month after the final dose of the vaccine. So considering that we have these 15 vaccines authorized around the world, there's actually a paucity of data on how good they actually are. So in terms of the US, I think we have potential EUAs in 2021 for AstraZeneca at some time. The trial's been done as of March, end of March, but nothing has happened. I heard about it going to the FDA. Novavax have indicated their protein-based vaccine will go for approval to the FDA to get EUA in the third quarter of this year. The Novavax vaccine is this recombinant protein-based vaccine that is based on protein plus adjuvant, their proprietary matrix M1 adjuvant. It's produced in insect cells. It's given as a 2-dose vaccine 21 days apart. They've done a number of phase 3 studies, and actually their phase 3 study they did in the UK was actually a very important study because they happened to compare their vaccine efficacy for the UK variant against the Wuhan variant, on which all the vaccines are based. And they found, very importantly, that the Wuhan-based vaccines protect against the UK variant. And that's really very important. So in the UK, it was 96% efficacious against the Wuhan variant and 86.3% against the UK variant. And in this country, we have about 70% of the viruses found in this country are the UK variant. So it's very fortuitous that we have vaccines based on Wuhan used in this country, which protect against the UK variant very effectively. The Novavax study also did a trial in South Africa, a small phase 2b study. And this was one of the first ones in the public domain to show that the vaccines against Wuhan strain may not be effective against different variants. In the case of this vaccine, it had only 55.4% efficacy against the South African strain in HIV negative people. If it was HIV positive people, the efficacy was very, very low. However, it should be remembered this is a very small study, only 4,000 people, and 1/3 of the patients enrolled were already seropositive against the virus, which complicates actual interpretation. And so we must remember this cohort is small, but it did make the suggestion that some vaccines may not be efficacious against all the variants circulating. So this brings us into the variants. Most of the variants are based on mutations in the receptor binding domain, which is, as the name implies, binds to the cell receptor, ACE2. And 3 separate mutations have actually been considered to be very important ones. And that is the D614G, which is the initial substitution identified in the United States and associated with enhanced transmissibility, but didn't seem to affect neutralization. The N501Y substitution was in the UK variants. And of course, we know, although it has increased transmissibility, the vaccines work against it. The E484G or Q is associated with the South Africa variant, and it appears to encode resistance to neutralizing antibodies made by the Wuhan vaccines. There are also some other mutations outside the RBD, but we're not going to have time to talk about those. So this resulted in the WHO coming up with a classification of genetic variants. And their way of looking at it was there's a lot of genetic sequencing going on, and this would be a signal to identify new mutants. And if these mutants started to have a selective advantage, they would become variants of interest. where the same mutant is identified in one or more geographic areas, but there was no fundamental change in the virus. And then if it really got going, we then get into the variants of concern. And these are mutants that have increased transmissibility. They cause more severe disease, potentially. They don't, vaccines don't prevent against clinical disease, and the virus is less neutralized by vaccine antibodies. So far, no variant has been identified that causes more severe disease. It's all been about transmissibility and the ability of the vaccines to work against the variants.
Alan,
Yeah.
about 3 minutes, please.
Maybe
Sorry.
slightly more than that, 4
Okay.
or 5, but yes, I'll do my best. The CDC came up with their own way of looking at variants, and they classified them into variants of interest, variants of concern, and variants of high consequence. And we have no variants of high consequence in the United States. We have a number of interests, New York, Brazil, and India, and we have variants of concern, the United Kingdom, Brazil, P1, South Africa, and a couple in California. But as I said earlier, at the moment, the UK variant is dominant in the United States, and the vaccine works against it. So we're in a good scenario at the current time. In terms of the variants of concern, the UK variant has a number of mutations, of which 501 is the most important. It has this increased transmissibility, slight reduction in neutralization, but the vaccines work against it. When you get to South Africa, we have more mutations, we have increased transmissibility, neutralization is reduced, and we have reduced efficacy by a number of Wuhan vaccines, but not all of them. And so the Pfizer vaccine works against the South Africa variant, for example, and Moderna have shown that they can boost with a South Africa variant and get very good immunity. The Brazil variant appears to be one also increased transmissibility, decreased neutralization, but there's very little data to actually say in concrete what is going on with that variant and how different vaccines work against it. But there appears to be evidence of reinfection for those after the Wuhan variants and had a primary infection. The one I'm most concerned at the moment is the India variant, and this is a so-called B1.617. It has two separate mutations, 484 and 452, but actually it's 4 different variants. The initial 617, and then it's evolved into 3 other variants, where you can see here on this slide has many more mutations. The evidence to date suggests the Indian variants are much more transmissible than the other variants identified to date. But the Pfizer vaccine, the only one in the public domain, is said to have 100% efficacy against the Indian variant. This slide here shows the different variants, and I'm showing this slide to make the point that the mutations are not random. If you look at the spike protein, there's only about a dozen sites that are actually getting mutated. All the rest of the sequences are homogeneous. And so I think this is an argument that the virus is going through convergent evolution, and we can talk more about that later. But essentially, the mutations are happening at the same places in different countries, and we're heading towards a strain that will be, hopefully, and I'm being optimistic here, the basis of one vaccine that works against all variants. Where does this action happen? It's on the spike protein, it's on the upper face that interacts with the ACE2 cell receptor. And so the idea is that these mutations allow tighter binding of the spike protein against the ACE2 receptor, and that's why these viruses are more transmissible. So where we are at the moment in the United States is the UK variant is 50% more transmissible than the Wuhan variant. The Indian variant appears to be 50% more transmissible than the UK variant. That's what's stated, although the evidence is not strong. The UK variant makes up 70% of the virus detected in the US, and so I think we're good with the Wuhan vaccines. However, the Indian variant is now starting to be detected in the US, but is less than 2% of the variants identified to date. And it's unclear what will happen in the future. I'm going to try and be an optimist here and say I'm hoping the UK variant will stay dominant in the population as it has done against South Africa, Brazil, and other variants to become the predominant virus, and I hope it keeps that way. Clinical trials have changed. I think that's the big message moving forward. Originally, we looked at vaccine efficacy by looking at reduction of clinical disease in vaccinees versus placebos. Now, every clinical trial has to analyze every case of COVID by RT-PCR to identify the particular variant. And now we look at vaccine efficacy in terms of the vaccine efficacy against a particular genetic variant in a particular geographic area where the clinical trial site is. We have a lot of studies showing vaccine sera neutralize genetic variants differently. But at the beginning, all the evidence so far is that neutralizing antibodies are not a correlate protection. And so I think we have to be careful how we analyze the neutralization data. So where are we now moving forward? The current vaccines are based on the Wuhan genetic variant. There's limited data available for the efficacy of most of these 15 vaccines around the world against genetic variants. I believe there's convergent evolution of SARS-CoV-2. I should say, I hope there's a convergent evolution that will can have one vaccine that will work against all variants. A big complication at the moment of doing COVID vaccine work is having 8 different neutralization assays in use. It really makes comparison of data between different studies very difficult. The WHO has identified an antibody standard, but the real question is, will people use it? Those infected with SARS-CoV-2 appear to have more severe disease as you get older. The immune response is better, whereas if you have the vaccine, the immune response tends to be better if you're younger, but still protective if you're older, even though you don't have high titers of antibodies. The period of time between doses 1 and 2 is an active area of research based on the initial AstraZeneca studies in the UK. A big question is now, how long will immunity last, and do we need booster doses? And the other big thing that's happening at the moment is something called mix and match, where there's a number of clinical trials going on using heterologous prime boost and boosters to work out what can you mix different platforms on the first and second dose with boosters to actually increase the immune response and longer term protection. So I've covered an awful lot and I've gone over time slightly, Stan, I apologize. There's an awful lot of information to get across in a very short period of time. Thank you very much.
So as we do here, we'll turn it and open it up for kind of questions to the both of you, either for a discussion or if one question is more suited for one of you, for that individual. I do have, like I say, a desk now of notes of questions, but I'm going to open it up to the floor. There were questions submitted in advance, but if you did submit a question in advance, why don't you go ahead and unmute yourself and put your video on and ask it to our guests. Don't be shy. Well, all right, then I will start off. I'll take the host. The host. I've kind of a softball question to start off. All right. So this will be for both Alan and Roger. So again, pleasant surprises in the last year. You touched on some of them. If you could both reiterate, and unpleasant surprises in the last year, what you never thought would bite you on the backside.
Alan, you want me to go first?
Sure, go for it.
All right. So a pleasant surprise was clearly the efficacy. I mean, there were a lot of surprises in terms of how quickly we were able to move, but I think some of those were planned, or at least we anticipated that we could move things forward quickly. The efficacy of the vaccine was just a huge and exciting win for us. I don't think in my career, I've been in the industry for 40 years, I don't think in my career I've been that excited about something. To see 95% efficacy, that was just extraordinary, and it really, for those of us who had been working tirelessly for a long time, it really geared us up to keep going, because now it was time to really make the Doritos faster, basically. In terms of things that we didn't expect, there were some glitches, and I can't go into a lot of detail on some of the glitches, but most of them were relatively minor. But in some cases, when you have uncertainty with a glitch because you haven't done a sufficient, I would say a sufficient prospective development program, you have to go back and do retrospective work. And so that becomes a little bit unnerving because you're not entirely sure what you're going to find when you start digging back into your process. Normally, what we like to do with a conventional program is do prospective development, meaning we try to understand the process and the product very, very well, so we don't have any unanticipated, unpredictable problems. But there were, because of the nature of the way we did this, the parallel development in particular, there were a number of little glitches that popped up that we had to do some retrospective work, and that That required a huge amount of time and effort in a very limited framework because we were still on a time clock to get this out the door.
Yeah, I'd agree with what Roger said. To me, the most amazing thing for the whole process has been the collegiality between everybody. People who traditionally compete with each other have been very supportive of each other. Everybody wanted to work together and get the vaccine. I think everybody realized it was a pandemic. It wasn't about profits. It was all about getting vaccines out there as quick as possible. And everyone worked together. It's been, I think, an amazing experience seeing what's happened. Even regulators, and to follow what Roger was saying, the FDA, to me, with their guidance documents, they could produce, what, 3 or 4 of those. They were very explicit, long. They told you exactly what they wanted to get to go into the EUA. It was very impressive what they managed to do and get out to everybody. I think it's been a phenomenal year and a half going through this. I think in terms of the things, just as Roger was talking about, the manufacturing's had its ups and downs. We've done a lot of clinical trials, and I can tell you that's equally the expectations of doing the clinical trials has been, I've never seen anything like it before, where we do trials for this, and essentially you do your clinical trial during the day, and then you have to do your quality assurance in the evening, and then before you go to bed you have to upload the data to the sponsor, be it Pfizer or be it NIH, whoever it is. And then by the time you get up in the morning, then all the queries have to be answered by 10 a.m. before you start the next day's volunteers. And that's been happening continuously. And I think it's amazing what people have managed to do to keep up with all these clinical trials. It's been phenomenal. Everyone doing the amount of effort put into these things is phenomenal. Normally, when you do a clinical trial, you're providing data to the sponsor every month, but this has been every night.
Yeah.
And because everyone wants to know in real time what the data's like, what COVID cases are coming through for VE. Are there problems with AEs? I mean, it's really been phenomenal the amount of pace that's been done to go through this. Probably next thing I'm going to say probably is not going to be popular with politicians. I feel that we, in some ways, have been our own worst enemies. I mean, I've been on presentations now where politicians say, "Oh, we know those scientists can make vaccines in a year," and I just don't think they understand the amount of effort that's been put in to deliver it. Now they say, oh, it's not a matter whether the scientists can make the vaccine, it's just a matter of whether we can deliver it to all different countries. And it's just not fair on the amount of work that's been put in to get those goals. And I think now there's this expectation that the next pandemic or the next vaccine can be done in a year. And I just don't think the pace is sustainable, what's been done in the last year.
I fully agree. I fully agree.
I'm not sure how we, as scientists, moving forward, how we cope with that. I think it's going to be a problem when the next infectious disease comes along. They're going to say, "Oh, those scientists can do it in a year," and that concerns me.
Yeah, Alan, it's not just politicians. Internally in my company, that's what they're saying.
I'm not going to comment on that one.
Well, and you know why? Because
yes.
You've done it once. So
yeah.
why can't you
yeah.
continue to do it?
Yeah.
We'll give you more money to do it. Well, it's not about more money.
No. Yeah.
So,
and to me, I really think it's been great how everybody's worked together. I mean, it's been phenomenal seeing
absolutely.
traditional entities who compete working together.
Do you think this will come back and bite you? You know, let's say it's not a global pandemic, but why don't we have a good TB vaccine? Is that because Pfizer doesn't want to help people in India where you have a million deaths? What about a good HIV vaccine? Is that because that bad Pfizer group doesn't want to help HIV compromised individuals? Do you think that kind of pushback is going to come?
Actually, it's gone the other way, Stan. So we have more interest in doing that kind of work now. But the problem is that what Alan described, the sustainability of the speed with which they're looking for it. Because there's not if it takes 6 years to deliver it, it costs that much more money and it's that much more time, and you're distracting from other things potentially. And that balance in the industry is always going to be there. There's always going to be a tension between making money and delivering, good, meaningful medications. And I'm fortunate because I sit on the scientific side. I don't have to make those commercial decisions, but I have to deal with those people. And, it's a bit of tension, and the tension, like it or not, plays out in all sorts of ways.
Yep.
I'll give you another example, and this one came to me after Alan started talking. One of the things with the clinical trials that was really critical for us, and I know for Moderna and many companies now, is making sure we had the right level of diversity in the cohorts, because that for a lot of reasons, but one was there are certain cohorts that would be against taking a vaccine like this because they don't trust it, and particularly in the U.S. And so, we worked really hard as an industry together to try to figure out how to maximize the diversity in our trials, so that it was consistent with the demographics as much as possible. And I think that's something for the future that's going to be extraordinarily important for all clinical trials.
Okay. So I'm going to read a few questions that had come in, unless someone wants to unmute and ask a question. Give you a second. All right, so a question that came in was, how long is the vaccine valid for in the body? And I guess you kind of alluded to this. In other words, is your best guess yearly boosters, or is it still way too early and you're not going to speculate?
I don't have a right answer for this. I think the data currently shows that we have pretty good durability through 6 months. We'll be monitoring people, for the next 2 years at least. And different populations may behave differently in this space as well. So we're gearing up with the thinking that we're going to need some sort of booster or some sort of routine COVID-19 vaccine. But we don't know whether or not it's going to be absolutely necessary or to what extent it will be necessary. A lot depends on the strains. Alan identified a number of strains. So far, we've been very fortunate that most of the strains, if they are getting convergent, maybe COVID-19 will wear itself out. But who knows? Things change all the time.
Yeah. In terms of crystal ball gazing, and I have no evidence to support this. Okay, this is just an opinion. I want to stress that. I would not be surprised if we did a COVID booster in September, October, when people get their annual flu shot and we know they're coming back in to get the flu shot. It is a real opportunity to give people a COVID vaccine at the same time.
Okay.
And if it was me and I was planning, and it's not me, of course, I would think about doing that, because you've got a captive market coming in for their flu vaccine. Getting people back as repeats for vaccines is incredibly hard. We all know that. And it's real opportunity, I think, to give people the COVID with the flu vaccine, to give protection across the fall. That's my guess. And I have no evidence to support that. Okay, well, you make that very clear. That's just an opinion of what I would do.
All right. Thank you. Bernardo Reyes, you had a question, comment and question in the chat. I'd like you to unmute, and if you don't mind asking it.
Yes, sure.
Thank you.
From the studies of the mix and match vaccines, higher antibodies values have been reported from the heterologous vaccination than the homologous one, even against the variants. And could this mean that this method will be used from the development of future vaccines?
Who's going first, you or me, Roger?
It's your turn.
Well, I think the mix and match studies are actually ongoing now. I'm not sure I actually believe there's data in the public domain on the clinical trial to support that. I think there's a lot of in vitro work, but not much done from the real clinical trials. And I made a point on my last slide. There's 8 neutralization tests being currently used, and they all give very different values. And trying to interpret those values is really very hard. Just because somebody has a bigger number than somebody else doesn't necessarily mean that's right. And I'll use an example. Novavax have the largest numerical figures for neutralization titers of all of them, but their convalescent plasma are higher than, say, a Pfizer or a Moderna study. And so their assays are more sensitive. So does that mean their vaccine makes more antibodies than somebody else's? I just don't think we know. there's a real paucity of data in terms of comparing the neutralization assays used with different vaccines and trying to make sense of it in terms of humans. I think it's very easy to do the studies in cell culture, in vitro, with the reporter assays, etc. But trying to translate that into VE is very hard. And as I said earlier at the beginning, I think that's one of the big pushes, what we call the immunobridging studies now, to try and work out, can we try and correlate a certain amount of VE with a particular biomarker? But at the moment, I don't think we know what that is, be it cell receptor in competition assays, spike ELISA assays, or a neutralization assay, live virus or pseudovirus, or reporter assays. I just don't think we have that information to say that. It's ongoing now, but I really think it's going to be the vaccine efficacy studies that will give the information to support those biomarkers as we move forward.
Neha, you had a question in the chat, though it was a direct message. Neha, if you wouldn't mind, there, I see you on my screen. Could you ask your question? It's a great question also, just like the last question.
Sure. I always thought about, I assume the safety studies cleared this, but because these are messenger RNA vaccines, and I understand they have a short life, and but are there studies that looked at what host cells take up these messenger RNA and possibly express that as a protein, which might be then targeted by immune cells? In other words, is there some host cell lysis observed with these messenger RNA vaccines?
Over to you, Roger, first.
I'm not sure how to answer this one. I'm not sure I'm qualified to answer this one. You're probably in a better place than I am.
I'm hesitating because that information I don't think is in the public domain. That's why I'm hesitating about answering it. Studies have been done to look at that, but I don't believe they've appeared in peer-reviewed papers. There's a little bit appeared. I think there was a paper in New England Journal about following some people who had a messenger RNA vaccine, about where it was biodistribution, but a lot of work has not appeared in the public domain yet, and I feel I can't talk about that, I'm afraid. It's inappropriate, I'm sorry. I did give a disclaimer to Stan at the beginning. There are certain things that if they're not in the public domain, we can't really talk about it. And that's one of them, I'm afraid.
Can I ask a variant of Nia's question that may or may not? So
yes, Your Honor.
is this great antibody or efficacy response that you're seeing from the mRNA vaccines, do you think it is because there may be a combination of a T cell response from the peptides being processed from the newly translated protein, in addition to perhaps a B cell activation from the intact protein being presented on the surface? So that might be a bastardizing Nia's question, but do you think we're seeing kind of both of those pathways that you may not see in an activated vaccine, perhaps?
Roger's nodding there, so that's good.
I mean, again, we don't have the data in a place where we can publish it, but I would say that it's moving in that direction. How's
that? I think it's pretty clear that the way the spike protein is presented is very different for the different vaccine platforms. And how that exactly relates to the immune system, I think is very complicated. And I think we have a lot to learn about that. And for example, why do apparently the adeno and chimp adeno vectored vaccines associated with the thrombosis, whereas the messenger RNA vaccines are not? And there's been a lot of hand waving to suggest why that takes place, but I don't think we have the evidence to support it. I think seeing the data that's presented in papers on ELISAs versus receptor assay, competition assays versus newts, qualitatively, I said at the very beginning, qualitatively, all the vaccines are very different. even if you take the adjuvants, some of the papers that have compared multiple adjuvants, there's one by Balipalundran in Nature, I think it compares about 7 adjuvants. And it's really quite interesting that the quality of those immune responses are very different between the different adjuvants, even though it's using the same platform. And as I said at the beginning, some of the inactivated vaccines are using 2 adjuvants. And so it gets confusing how you actually interpret that to give what I call a paintbrush answer. And that's why I think of it as being qualitatively, each vaccine is different qualitatively. And do we know why the mRNA vaccines give higher VE than the other ones at the moment? I don't think we can give a categorical answer to that at the moment.
Okay, thank you. I'm going to ask Torek. He has a question in the chat. Torek, he or she, I apologize. Could you unmute and ask your question?
Yeah, I was just wondering whether combining this with a flu vaccine would make it much more expensive, or you don't think so? I don't know if you are.
That's got to be a Roger question.
There's always a cost. I don't know I don't know if it'll be much more expensive. I mean, we're getting to the point now where like I said, we're kind of making the Doritos. It's getting a lot more efficient. We are understanding more, and when you combine it, there's another question. I think Melissa asked a question about whether or not we'd have to run additional trials. The answer to that is probably not. Or if we do, it's going to be comparability work that we have to do more than anything else, because we get, for the flu, the annual flu, you get the northern and southern hemisphere projections of which variants or strains that you're going to work with. There's already a fair amount of data to substantiate, how to move forward, and so the studies are relatively quick and dirty to be able to evaluate that. And with COVID, if it's the same platform, the platform's already been tested. So it's really more the comparability of including the 2 together in one vial or one syringe.
The CDC have said that they don't mind what vaccines are included now when you get your COVID. There's no differentiation, which there was earlier, which I think opens the door for giving COVID and flu vaccine together in the fall.
Yeah, I mean, that's almost a, rolling off the log for us, if we can do that, because it's not a big stretch, and it will be an mRNA. For us, would be an mRNA platform because we've already got it down pat.
So, Torek, does that answer your question? Melissa, does that also answer your follow-up question?
Yes.
Yes.
Okay, thank you both. Young Lee, you had a question in the chat.
Yes, I have a question about the DNA vaccine by Inovio. As you know, the DoD pulled the funding for the Inovio DNA vaccine phase 3 trial. So why this vaccine seems to fail to meet the endpoint, especially the neutralizing antibody?
I don't think we know it's failed because there's been no peer-reviewed publication on the clinical trials with that vaccine. All we know is what we hear in the media, and I think it's very hard to read through and use media information. Apparently, the funding for the Inovio vaccine, their sponsor is not giving money for the phase 3, and so they are hunting to try and find more support for that, and I don't know why that is. It hasn't been in the public domain, the reason why it stopped, nor has there been a publication to explain why that took place. I think we have to be very careful taking the media, and I'll use an example, if you don't mind, with Pfizer, and I always quote this one, that quarterly you get these financial reports. And Pfizer gave a presentation on their vaccine. It had 26 slides. It was hugely technical about use and manufacturing. But one slide was about adverse events. So what got reported out of the 26 slides from the meeting? It was the adverse events, because it was the only slide the reporter understood. And so it was said, Pfizer vaccine causes lots of adverse events. Well, that wasn't true at all. But it was the only slide that the person understood. Because these financial meetings are meant for advice in the stock market. They're not meant for reporters. and the reporters don't understand it. And I think we have to be very careful what we read in the media and how we interpret it. Because reading what someone writes, and every reporter tries to give a different angle, and I think it's very hard to read those articles and be able to get information out that is actually usable. It has to be taken with extreme care. Roger, do you want to answer that one or not?
Well, I was thinking of a similar example in a different way. I don't know anything about the Inovio DNA vaccine, so I can't respond to that. But just in general, again, your comment about the media. There were two platforms with the COVID vaccine. One platform was the science and regulatory platform that we were all trying to push through to get approvals and make sure we had the right science in place and gear up for additional studies. And the other was more the commercialization and supply chain. And you're working with two different government bodies, and a lot of reporters conflated the two or got them mixed up. And it's relatively complicated when you sign a contract with a government body. You're making an agreement to provide a certain amount of inventory over a certain period of time, and you're allowing them to take control of it. And so in some countries, there were different expectations. And I know in India, we actually approached India early because we thought they could benefit from the vaccine, and we wanted to get a contract to be able to deliver the vaccine. And they put up a bunch of obstacles for us. And we said we can't address those obstacles. They want us to do a separate clinical trial with 36,000 patients or subjects, and we said that would be a waste of time. We've already got the data. You need to work with the data we have. And so, but the way it got reported back was, big bad Pfizer doesn't want to provide a vaccine to India, and then there was talk of, well, let's waive the IP for the vaccines and maybe transfer that to different countries. India has the largest vaccine manufacturing network in the world. Certainly they could manufacture for themselves. Do we just share our IP with them? Well, that becomes now again that tension between the business and the science. So and those things get reported out again as industry bad, everybody else good.
I think we're going to go with industry good in this case, okay?
Thanks, I like that.
All right, so there's a question that was asked earlier or mailed to us. Could you comment on kind of the concept of one herd immunity, maybe very quickly what the heck you think it is, especially maybe something that I could understand, and two, will we get there? And I know Alan's thinking, God, what the hell with this water, which understand very little, Alan. Thank you. So herd immunity, are we going to get there? Do we need to get there? What are your thoughts on that? Or is this just again, a media thing?
I guess I got that one as well, didn't I?
I think so.
I think the idea of herd immunity is very important, because if you have sufficient number of people immunized or had the natural infection, then the virus has a new host to go to. And in support of that is the nice studies on the vaccine showing that if you're vaccinated, you don't get infected, you don't excrete virus, you don't transmit it. So the vaccine is giving you good protection. Of course, we don't know how long it's for. And herd immunity is calculated based on R0 values, the reproductive number of the agent. And as you know, politicians got big into R0s. If you bring your R0 down, then you're doing well. But of course, if the R0's changing, then also it changes in the percentage of the population that need to be immunized to get protection for everybody. So if you think of measles, it has an R0 of around about 18. So you have to get 95% plus people of the population immunized. But if the R0 goes down, then it's the chance of infecting other people is reduced. Therefore, it's not, herd immunity isn't the higher, the number of percentage of the population need to be immunized is not so high. I think the way I think of it at the moment is convergent evolution. Is this, if we can get to one virus where we can make a vaccine against it, then I think we're on for herd immunity. I think as long as there's variants around where the vaccines have variable protective abilities, depending which platform it is, sort of there's qualitative differences. I think talking herd immunity is not so straightforward, because it will depend on which vaccine you take to prevent which variant. And at the moment, I still think that's complicated. And the way I tend to look at it at the moment is I'm a happy person because the UK variant is outcompeting the Brazil, South Africa variant, etc. in the United States. And we have a vaccine that protects against the UK variant. So I think it ticks the boxes at the moment. And I feel quite hopeful about that. If something like the India variant comes in, or a different version of it, 716.4 or 5, and the vaccine doesn't work, then I think there's a different game altogether. But for the moment, I think we're doing okay. I think we've done pretty well immunizing all the people we have. If the R0 keeps coming down as it is at the moment, then I think we're in pretty good shape. And herd immunity has a different definition as compared to if it's a huge outbreak where we have no protection against it.
The other option is to say, I mean, herd immunity is to protect the general population. If you want to say, as long as the vaccine protects those who want it, you know, is that good enough?
So I said earlier that I have this hope that we can give the vaccine to all school-age children.
Right.
And I think if we can get EUAs for the vaccine to give it to all school-age children, then I think that's going to be really good for the country, because the children who go to daycare and they do the schools, they're the ones who are infecting people. And I think if we can pull that down, then I think we are on the way to moving forward. Studies in the UK show that the people who don't get vaccinated are the ones who make the variants. You can actually detect variants in the people being developed. So there is a good push to vaccinate, although I'm not sure how you translate that. But I do feel if we could immunize all the children, get everybody back to school without the risk of it causing outbreaks, then I think that would really be a big plus for everybody.
And Raj, from your perspective, any crystal ball on when you might expect emergency use down to 6 months? Or is that because I know you guys are doing those studies, right?
Yeah, I mean, a lot depends on enrollment, how quickly.
Sure.
The amazing thing though is the enrollment for children has been outstanding. We have people lining up. So for the reasons that Alan indicated, everybody wants the kids to go back to school. They want them to be protected. So my hope is that that won't be a particularly big issue. The safety profile has been really good for the these mRNA vaccines in particular, but the other vaccines as well. And I would just hope that would be quicker rather than slower. I just can't predict when, because these studies are being run a little differently than we ran the initial study. It's not the same pace, even though we're trying to get most of the data wrapped up as quickly as we can. Again, diversity plays a big role, and we want to make sure we have a good demographic of folks in that diversity cohort.
Okay. And we definitely appreciate your work. One final question, and I'm going to, unless someone unmutes and asks, I'll ask kind of one final question. Anyone? Here's your chance. All right. So I've definitely heard from a number of presidents of medical institutions, the Fred Hutch, et cetera, that they will look to require vaccinations once we move from emergency use to actual licensing because of some legal considerations. Can you just educate me on when and what might be necessary to kind of, in this country, move from the emergency use authorization for adults to full licensing? How is that going? What's left to do?
So Pfizer announced that they filed, our BLA in May, and Moderna, I think, just announced that they filed their BLA and just recently. Yep. We don't, it's unpredictable, but it's between 4 to 8 months by the time we get approval. And so what happens then is once you get approval for the BLA, the EUA doesn't necessarily go away, but it sort of diminishes. You can't keep sending stuff into it because it's kind of like, the EUA is kind of like this very flexible place where you can send a lot of information and get it approved very quickly. Once you have the license, it's a little more formalized. And the regulators want that, obviously, because that allows them to manage that a little bit easier. And essentially what we've done is we've converted all of our information into what would ostensibly be the licensed information anyway. So any time between now and say, early next year, or the end of this year, possibly, we could, foresee the possibility of an approval for a license in the US.
So Stan, I just need to temper that by saying that once you have your license, doesn't mean it will get used, because CDC, ACIP has to formally approve the use of the vaccine to be implemented in the United States.
That's right.
Unless people buy it privately. This is a big deal for insurance. If you haven't got a CDC approval, then normally you can't get a vaccine on insurance.
That's exactly right.
And so that's why we keep seeing the FDA do the EUA, and then ACIP comes in and then follows up with a concurrence recommendation, etc. And so it's going to be part of that process. Just having the BLA is not going to be enough. I think ACIP will work very fast to give an approval, but they don't really do anything until the BLA has been approved. And the FDA don't normally talk about the BLA until it's actually been approved.
Okay, and so soon, but not immediate. Let's go with that.
Fair enough. Fair enough.
Well, with that, I'd like to thank both of you again for, really, superhuman work this last year. And unfortunately, I suspect it's going to continue for a while, but we again appreciate you taking the time out of your busy schedules to talk with us. And again, many, many thanks to you, your colleagues, the teams you work with. Very much appreciated. So please be safe, and thanks again.
Thanks for
Thanks,
the
Stan. It's been a
opportunity,
pleasure.
Stan. Thanks. Alan, good to talk with you again.
Good to talk to you, Roger.
Take care.
Okay, bye-bye.